Enzymes increase the rate of chemical reactions by decreasing the activation energy of the reactions. This is achieved primarily by the enzyme preferential binding to the transition state of substrate. This model is proposed by Linus Pauling in 1946. Catalytic groups of enzymes are required to achieve a specific reaction path, the conversion of a substrate to the product. In this chapter, we shall illustrate some fundamental aspect of enzyme catalysis using an example, the serine proteinases, a group of enzymes that hydrolyze peptide bond in proteins. We also examine how the transition state is stabilized in this particular case. Higher organisms like us use proteinases for such different functions as food digestion, cleavage of signal peptides, and control of blood pressure and blood clotting. Lower organisms like bacteria also produce many different extracellular proteinases to degrade proteins in their environment. Trypsin, chymotrypsin, elastase, and subtilisins belong to serine proteinases. We shall define some basic enzymological concept. BKLS and Mantan laid the foundation for enzyme kinetics as early as 1913 by proposing the following reaction scheme. Enzyme and substrate first reversibly combine to form enzyme substrate complex, ES complex, and then giving in second step the product. The reaction rate is a function of the substrate concentration following hyperbolic curve as shown in this picture, and then this could be explained by the equation, and from this equation could be utilized to derive this Michaelis-Menten equation. This Michaelis-Menten equation has two parameters, Vmax and Km. Vmax is going to be Kcat times enzyme total concentration, where Kcat is called turnover number. So it could be defined as the Vmax divided by enzyme concentration. Vmax is the catalytic activity at very high concentration of substrate. K is expressed by using this equation, where you can see the reaction constant for three reactions. And understanding the Km is a little bit complicated, but I'm going to just mention two simple concepts. One, this could define as this equation. So looking first reaction step, that could represent dissociation constant of ES complex. So the smaller, the high affinity between two complex. Second meaning of Km value is the substrate concentration, where the half maximum the reaction rate reaches. So the smaller, again, it is more efficient in catalytic efficiency. Third concept, Kcat over Km, called catalytic efficiency, could be defined as initial velocity over substrate concentration times enzyme concentration. Catalytic efficiency is the single most important parameter for enzyme. It could represent rate constant that refers to the overall conversion of substrate into product. An enzyme increases the reaction rate by binding and stabilizing the transition state of its specific substrate tighter than the ground state, called transition state model. In this diagram, the high activation energy in uncatalyzed reaction in A, the catalyzed reaction has a lower activation energy. This could accelerate reaction rate. Both reactions proceed, A and B, through one or several transition states. Only one transition state is shown in A, whereas the two bumps in B represent two different transition states. The decrease of activation energy could be achieved by a variety of ways. The most important one is that the enzyme could make its complex with consuming low energy compared to its normal state or ground state. This one or this one. The higher affinity of the enzyme for the transition state makes the transition state energetically favorable and thus decreases the activation energy. This is just the wild type enzyme, B, the mutant, where the transition state is much stabilized, so the reaction may go faster than wild type. If, on the other hand, the enzyme were to bind the unaltered substrate more strongly than the transition state, as you see in C, its complex is much tighter than the wild type. The decrease in binding energy on the formation of the transition state would increase, as you see the larger scale than the wild type. So it would increase the activation energy and catalysis would not be achieved. Now we move to the serine proteinase. Serine proteinases catalyze the hydrolysis of peptide bonds within polypeptide chain. The bond that is cleaved is called scissor bond. RAX, RAB represent polypeptide chains of varying lengths. The reaction proceeds in two steps. The first step produces covalent bond between C1 of the substrate and the hydroxyl group of reactive serine residue of enzyme. Production of this acyl enzyme intermediate proceeds through negatively charged transition state intermediate where the bond of C1 have tetrahedral geometry in contrast to the planar triangular geometry in peptide bonds. This structure is called transition state. This one is clipped. Next step, one peptide product is attached to the enzyme in the acyl enzyme intermediate, and the other peptide product rapidly diffuse away. Second half at lower bottom is essentially the reverse of the first half. The acyl enzyme intermediate is hydrolyzed by water molecule to release the second peptide product with a complete carboxy terminus and to restore the serine hydroxyl of enzyme. This step also proceeds through negatively charged tetrahedral transition state intermediate. Then, what are the structural requirements for the enzyme to perform this reaction, especially regarding transition state? This slide shows a schematic view of the presumed binding mode of the tetrahedral transition state intermediate for the deacylation step in the active site of the serine proteinase. The serine proteinases have four important structural features in common. In yellow color, catalytic triad, oxyanion hole, specificity pocket, main chain substrate binding. I'm going to explain the role of each component. First, catalytic triad. Catalytic triad consists of the side chains of serine, histidine, and aspartic acid close to each other. Serine residue is reactive group and forms a covalent bond with protein substrate shown here. To make such a tetrahedral intermediate, the initial state, so here you can see catalytic group serine make a hydrogen bond with the histidine, but soon the proton is transferred to the histidine so that OH is converted into oxyanion, negatively charged oxygen, which could attack carbon of carbonyl group in substrate peptide bond, red one, forming this kind of tetrahedral state. Protonated histidine is further stabilized by negatively charged aspartic acid, positioned in here, not shown in this picture. So, three residues working together cooperate to facilitate this intermediate.